Hi everyone, my name is David Eberts, and we are going to go over RNA interference. Let's start off real quickly with saying what type of experiment this is. This is a lose-it experiment. And you can kind of derive that from the name and the fact that it's interference. We're going to interfere with something, that means we're going to lose it. It's a lose-it type of experiment. It's a pretty simple concept, so we don't need to go into too much details of the actual techniques of it, but it works, of course, by targeting RNA. We'll start again by drawing our cell. Inside our cell we have our nucleus, and this is a cell, and inside of the nucleus we have DNA, and DNA is constantly being transcribed for any genes at all and making mRNA. And the mRNA will flow out into the cytoplasm, and from interacting with ribosomes, we'll call that a ribosome, will then lead to protein translation. We'll have a full-blown protein made. If you have any type of experiment where you want to see what would happen if we knock out, is another name for this, or silence a gene or protein. So this is for knocking out, or the same exact thing, you could just call it silence. Silencing. Silence is with an E, all right. And basically we're going to interrupt this pathway that is the central dogma of how cells work. Now outside of the cell in your lab, you are going to create a strand of RNA. And this strand of RNA is going to be very short. It's going to be called siRNA, and in its name is that word short. It's short interfering RNA, and it's very small, around 21 base pairs. And if you don't really know how many base pairs is a lot, 21 is very small. It's a very tiny base pair. So what we're going to do with our siRNA is we are going to inject it into the cell, and it's going to block this process from happening. That is where in the pathway it is going to cancel gene expression. It is going to knock out or silence our gene expression to make the protein. And the concept, once again, like I said, is very simple. So siRNA is put into the cell and blocks this from happening. Now let's look at how that is done. I'm just going to draw a giant nucleus, and you can imagine that the entire board is our cell. Why not? Inside our cell, we have mRNA being made from our host DNA, and the RNA will want to come out and be translated. And what's going to happen is, as mRNA is being made, we are actually going to break this. I apologize. I don't care about the nucleus right now. The mRNA being made is going to be very long. We talked about 21 base pairs being very short. Regular RNA is actually pretty long. It's huge. Hundreds of base pairs, if not thousands of base pairs. This is our mRNA. The siRNA is going to come in and bind to a complementary sequence. It's going to find a region on the mRNA that is complementary to our siRNA, meaning if this base is A, then this base is going to be T. If this base is G, then this base is going to be C. But I apologize, this is RNA, there's uracil, not thymine. And exactly what you think would happen is it'll come in and bind right to our DNA, right to our RNA, sorry. And so this is our siRNA. So our complementary sequence at our siRNA, we knew in the lab to target for exactly our gene, whatever gene interest this is. We'll call it gene X. We made a complementary strand bound to it, and essentially what we have now done is we have now created double-stranded RNA. dsRNA. And for those of you who can't think of it, double-stranded RNA is not a good thing. And like most things inside the cell, when there's something is there that there shouldn't be, it will destroy it. It will try to get rid of it. So that is exactly what the cell does. It'll come in with some sort of protein complex and slice up that double-stranded RNA. It'll come in and cut it and degrade it. And what will be left is, here's half of the RNA, pretend there's breakage, and here's the other half. And it's going to break right where our double-stranded is. So we'll have break, break, and now we have broken mRNA that's double-stranded, and this whole process will then call for both of these and all of it to go and get degraded. Degrade. So essentially, you are destroying all the mRNA of your gene X inside your cell. And with no mRNA being present, you will never get any protein expression, and you will successfully knock out or silence your gene. And then you can do this type of lucid experiment to see if something is necessary. You can see if gene X is necessary for me to have brown hair. You knock out the gene inside me. If my hair is not brown anymore, it was necessary. And that's basically RNA interference. Hope it helped. Thank you.